Hello students, Mrs. Klein's here with your next section of video notes over mutations or errors in the genetic code. A quick note before we start. There are many different types of mutations, but we are specifically in this section of notes addressing mutations that occur on one segment of DNA. So we're addressing specific gene mutations. In the next few units, we will start talking about chromosomal mutations which affect larger sections of DNA. So let's get started. What are mutations? Mutations are any change in an organism's DNA. This can occur in one specific gene or across an entire chromosome. Some mutations can affect the physical expression of a gene or its phenotype. That's an important vocabulary term to kind of commit to memory. Phenotype is the physical expression of a gene. For example, you may have a gene that codes for brown or blue eyes. The physical expression is your eye color. So some mutations can cause an early stop codon. We remember from translation how a stop codon will end protein synthesis. So let's say you have a mutation that occurs that codes for an early stop. Maybe it's a base swap that codes for an early stop. In that case, you may notice that the amino acid sequence stops early and the protein is incomplete. There are some mutations that don't really cause harm to a cell. Some mutations do not affect the amino acid sequence, and we call those silent mutations. You may remember from translation that some amino acids have multiple codons that code for them. So if your base mutation codes for the same amino acid, then there is no change to the protein structure. We call that a silent mutation. And there are also sections of DNA that don't code for a protein. So if those sections get mutated, then it may not cause problems either. And I think this is also a good thing to point out. Not all mutations are bad. Mutations have been occurring in DNA sequence of living things throughout history and essentially is going to be discussed as the driving force of genetic variation in our population, which can be a good thing. So let's talk about types of mutations. There are two major categories of mutations that I would like you to be familiar with. The first is called a point mutation, and the other is a frameshift mutation. A point mutation occurs when you just substitute one base for another. So you can see that in the image at the top to the right, instead of coding for thymine at the 5th nucleotide, the mutated strand codes for cytosine. So that will usually just affect one codon in the messenger RNA sequence if it codes for a protein. A frameshift mutation occurs when a base is added or deleted from the DNA sequence. We call those frameshift mutations because, as you know, codons are made of a 3-base segment. So frameshift mutations shift your entire codon sequence, and each codon codes for a specific amino acid. So that's going to affect many amino acids in the protein structure or polypeptide chain. Mutations, luckily, are relatively rare because there are different checkpoints built into the cell cycle to check for those errors. Remember that DNA polymerase is the proofreader of DNA replication, so it is also checking for mistakes in the code. So in the picture in the bottom right corner of your screen, you can see several checkpoints. If at any of those points there's anything wrong with the sequence, the cell may go into what we call apoptosis, which is programmed cell death so that mutation doesn't get passed on. So there are a couple different reasons why mutations occur. The first is whenever DNA is replicated during the synthesis phase of interphase, and the DNA fails to copy accurately. So a base may get substituted or added or deleted during this process, which will then affect every single cell after the fact. Because if that mutation makes its way through, the mutated DNA then copies during the synthesis phase of interphase, it will pass that mutation to its daughter cells. So sometimes these mutations can have pretty significant effects on the cell. Mutagens are external influences or environmental factors that can change DNA sequences. So there are some things that we know affect the structure of DNA. In the process of affecting the structure, it can weaken bonds between nucleotides or bases, and in that case, it can add nucleotides or delete them from the sequence. So radiation is an example, along with some chemicals like asbestos or nicotine, which have been known to affect DNA sequences in replication. Some of these mutagens can speed up the process of replication, which increases the chance that if mistakes are made, they are not caught. This also causes cells to reproduce more quickly than they should. Here is an image that shows you've got your double helix and then incoming UV radiation. And that radiation weakens the hydrogen bonds between base pairs and ultimately causes a mutation in your DNA sequence. When we think about cells that could be affected by mutations, there are two different types of cells to note. The first is a germ cell mutation. This is whenever a mutation occurs in a reproductive cell, so in a sperm cell or egg cell. The DNA sequence is altered, and in that case, the person that the mutation occurred in may not express any changes because it happened only in their reproductive cells. In their offspring, however, those offspring may inherit through sexual reproduction that mutated sperm or egg cell. Those are mutations that can be expressed by offspring or from one generation to the next. Next are somatic cells. Somatic cells are your body cells, and these occur within an individual, usually due to the environmental factor or a genetic factor that causes a mutation. But it really is only expressed in the individual it occurs in. An example would be a mutation that occurs in skin cells due to UV radiation exposure. That doesn't necessarily mean that the offspring are going to be affected, because it only affects the person's skin cells and not their reproductive cells. So let's look at how different mutation types affect a DNA sequence. In a substitution mutation, you've got one nucleotide that is substituted for another. This can have two different outcomes. You can end up with a silent mutation that does not change the amino acid sequence, or it can change the amino acid sequence, which alters the expression of the protein. So here is the original template strand of DNA. The messenger RNA is shown here in red and is going to be complementary to the DNA strand. The first codon codes for cysteine, using my codon chart or wheel, histidine, glutamine, and phenylalanine. This is my original DNA sequence. Let's see what happens whenever there is a mutation. Here on my template strand, my messenger RNA sequence is going to be changed from a C to a U whenever transcription takes place. But this codes for the same amino acid, so it will be a silent mutation. Now, let's say that original base is now substituted for a T. The codon on mRNA will now change to CAA, and now codes for the amino acid glutamine. This mutation changes the amino acid sequence, affecting the protein structure. An example of a condition that is caused by a substitution mutation is sickle cell anemia. Pictured is a normal cell DNA sequence and normal red blood cell. Now, if we have one mutation, it is going to affect the amino acid sequence. One small change in acid sequence leads to this, and cells being sickle-shaped because the protein inside the red blood cells changes as a result of that mutation. So sickle cell anemia is a condition that affects individuals. It can cause pain in extremities. It can cause people to feel very tired all the time because they are not getting enough oxygen delivered to their cells and tissues. You can see the shape of the cell is kind of a hook shape. This can cause blood clots to easily form, and it can also cause damage to tissues. This can become a life-threatening condition, all because of one small substitution mutation. Another example of a condition caused by a point mutation is neurofibromatosis. This is a benign condition, usually, but it may lead to other problems later in life depending on where these benign tumors form. So they are not all in the skin. Sometimes they form around vital organs, which can cause problems for the individuals who express this mutation. Another example is Tay-Sachs disease. This is a rare genetic disorder that gets passed from parent to child. So this affects the germline cells. It is caused by the absence of an enzyme that helps break down fatty substances in the nervous system. These fatty substances kind of build up to toxic levels in the brain and spinal cord, and it affects their function overall. Now let's look at a frameshift mutation. So in this case, a nucleotide may get inserted or deleted, which can cause the reading frame or codon frame to shift. Some of these may cause an early stop codon in the protein structure. So here is a DNA template sequence, which codes for this mRNA sequence in red and shows our base pairing rules. Using my codon chart, I can decode each codon from mRNA for the correct amino acid. This gives me cysteine, glutamine, glutamine, phenylalanine. Now, let's say this base in my template DNA strand is deleted. What happens? Well, that is going to affect my messenger RNA because that nucleotide will never be added. So it is going to shift all of these nucleotides in these codons to the left. Now my codon is going to include GTG, which codes for histidine. And my new codon here is TCA, which codes for serine. And then we'll lead to the next codon, AAC, which codes for leucine. So you can see many more of my amino acids are affected by a frameshift mutation than by a single substitution mutation. Here is another example: my template DNA sequence, my messenger RNA sequence, my original amino acid sequence. Now I have added a C nucleotide. This would be an insertion mutation. This cytosine is a new nucleotide. From that, my codons all shift from that point on. Here is my new mRNA sequence, and then these are my new amino acids that are coded for in the sequence. An example of a frameshift mutation can be seen here in the Belgian blue cow. This mutation that is expressed by this cow was caused by a frameshift mutation. You can see here that this mutation affects muscle growth. The mutation occurs in the myostatin gene, which controls muscle growth. So this one mutation leads to a 20% increase in muscle growth. And there are some farmers who intentionally breed this form of cattle for meat production because of that increase in muscle mass. That is the end of our video notes for this section.